Good morning, everyone, and welcome to the morning session on day 3 of the conference on the HPC applications. It is my pleasure to introduce our first presenter this morning. That's Dr. Rian Pierneef of the Agricultural Research Council. His background is that he is a researcher in bioinformatics at the Agricultural Research Council Biotechnology Platform. After completing undergraduate degrees in genetics and statistics, he entered the world of bioinformatics and obtained a PhD in bioinformatics from the University of Pretoria. Due to the cross-cutting nature of the ARC Biotechnology Platform, he is involved in numerous projects and research endeavors across the agricultural value chain. Currently, the most prominent research avenue is based on the surveillance and identification of foodborne pathogens using the next generation sequencing. And the title of the talk is also then Foodborne Disease Surveillance Using Next Generation Sequencing. Dr. Pierneef, please.
Good morning all. Thank you for attending this session, which I trust you will enjoy. My name is Rian Pierneef, and I am a bioinformatician, which means that I am a biologist
Just a moment while we sort out some technical difficulties.
Which I trust you will enjoy. My name is Rian Pierneef, and I am a bioinformatician, which means that I am a biologist and that I know just enough about computers to make me a danger in any HPC environment. I fondly remember an email thread where I kept on saying, "Please, sir, can I have some more calls? And I need more speed, Scotty." As always, the CHPC quickly assisted and asked for a scaling graph before unleashing me on the large queue. Thinking, how dare these guys question my super duper computing capabilities? I'm a qualified bioinformatician. I gathered the results only to realize that there was a piece of humble pie waiting for me. In fact, my runtimes were getting longer the more nodes I added. I should therefore start off by thanking the CHPC for all the assistance, support, and patient guidance through all my research endeavors, even when I'm demanding more CPUs when my code does not scale. That being said, let's start. In this talk, I will be discussing some of my research endeavors at the Agricultural Research Council Biotechnology Platform, which is focused on foodborne disease surveillance using next-generation sequencing. First, a bit of background on the ARC Biotechnology Platform. Located on the Agricultural Research Council's Onderstepoort Veterinary Research Campus, the Biotechnology Platform was established in 2010 as a major strategic priority of the ARC. The role of BTP is to create the high-throughput resources and technologies required for application in genomics, quantitative genetics, marker-assisted breeding, and bioinformatics within the agricultural sector. The focus of the BTP is to establish itself as both a research and service-driven institution, providing an environment in which highly skilled researchers can be hosted and trained. The technologies established within the platform are accessible as services to the ARC, collaborators, companies, science councils, and researchers across the continent. BTP is possibly one of the largest and most capitalized genomics facilities in South Africa. I joined BTP during the 2017-2018 South African listeriosis epidemic. This was the world's worst outbreak of listeria food poisoning, with roughly 1,000 confirmed cases and an estimated 200 deaths. This outbreak highlighted the vulnerability of the South African population with regards to foodborne diseases and greatly influenced my research activities. It became clear to me that the prevention of epidemics such as the South African listeriosis outbreak requires large foodborne disease surveillance and foodborne pathogen testing projects to protect public health and ensure that no lives are lost due to contaminated food. Recently, the World Health Organization encouraged countries to incorporate next-generation sequencing, in particular whole genome sequencing, in their foodborne disease surveillance and response systems. Management of foodborne disease threats requires the swift and correct identification of foodborne pathogens. Whole genome sequencing currently provides the highest possible resolution and strain discrimination for foodborne pathogens with a rapid turnaround time. The data generated by whole genome sequencing enables in silico determination of numerous critical aspects of foodborne disease surveillance, such as strain typing, antimicrobial resistance profiling, virulence characterization, and phylogenetic analysis. This negates the often cumbersome and time-consuming traditional typing methods, as whole genome sequencing is proving to be an all-encompassing method of foodborne disease surveillance. A practical example of the limitation of traditional methods would be the figure on the left. Traditional serotyping methods are only able to categorize listeria isolates to a serogroup level. This is indicated by the blue border. With respect to the South African listeriosis epidemic, sequence type 6 was of critical importance and is indicated by the red circle. As seen within the red border, sequence type 6 is part of serogroup 4B, yet there are numerous other types within the serogroup. As such, a higher resolution than offered by the traditional methods is required to correctly identify the specific sequence type associated with a listeria isolate. This is achieved by next-generation sequencing and bioinformatic analysis. An added bang for your buck when using next-generation sequencing is the identification of antimicrobial properties in foodborne pathogens. The drastic increase in the detection and prevalence of antimicrobial resistance is of great concern and needs to be addressed rapidly. The misusing and overusing of antibiotics cause bacteria to become resistant. This leads to common infections no longer being treatable and a broader human range, which culminates in longer hospital stays, higher medical costs, and more deaths. As such, the detection of antimicrobial resistant foodborne pathogens is critical in curbing the spread of resistance and the provision of safe foods. This is a schematic representation of a typical whole genome sequencing pipeline used in foodborne disease surveillance. After the sequencing data has been generated, quality assurance and control techniques are applied. Thereafter, parallel processes are used to ensure consistency and accuracy of the imputations. In the first process, the sequencing data is assembled using the brain graft techniques. The result is a de novo assembly of a bacterial genome. This information is then used in combination with reference databases to identify characteristics of an isolate, such as the species, strain, type, antimicrobial resistance, and virulence profile. The second process is based on mapping of the unassembled data or reads against the reference genomes and loci using a K-mer approach. The results from both processes are inspected for consistency and accuracy. Phylogenetic inference is then possible to ascertain the evolutionary history and relationship of an isolate with regards to other organisms. Sure, that seems quite complicated, you may think, but in essence, it's all relatively easy if you have computational power and memory. On the right is an example of what the typical raw output from a sequencing event looks like. It's just a file with a lot of letters and characters and includes the physical DNA sequence. The problem is that it is not a continuous sequence, but actually small pieces of the complete sequence. An analogy would be my wonderful kids, 2 copies of my favorite book and some scissors. A bacterial genome is similar to a book, only written in the letters A, C, G, and T. Awaking from a well-deserved nap, I find my ecstatic kids surgically dissecting the copies of my favorite book. I now need to try and reassemble the book from all the small pieces of paper. I have 2 options. One is in de novo assembly and the other is a mapping approach. For the de novo assembly, I would take all the pieces of paper and reassemble the book by looking for sentences that overlap. Because my kids are predictably random, I know that large portions of the book will not have been cut at the same place. As such, I would be able to overlap sentences and reconstruct a large portion of the book. The second option requires another copy of the book, which I can obtain from the internet. This copy will not be the same edition as my book, but I know that I will be able to reconstruct a large portion of my book by mapping the sentences to the internet copy. Got to love those kids though. Bacterial genomes range in size from a few 100,000 base pairs to more than 14 million base pairs. As per the previous slide, this is akin to a sentence consisting of A, C, G, and T with on average 5 million characters. The raw data per sample or isolate, usually obtained from a sequencing run, is roughly half a gig. This is before any QC or analysis has been done. After filtering and de novo assembly, this increases to 13 gigs. Again, this is for just one sample or isolate. Similar to the COVID-19 response, the only way to perform sufficient surveillance and curb the spread of foodborne disease is by testing, and more testing. Furthermore, this testing needs to be done across the entire farm to fork value chain. As such, we are talking about a lot of samples and data which needs to be analyzed in a speedy yet reliable manner. The box in the bottom right indicates the resources used for the de novo assembly of a single isolate. This is before any downstream analysis pertaining to identification, resistance, and virulence profiling, or phylogenetic analysis. It is therefore clear that a reliable and computationally proficient partner is required for any project relating to the surveillance of foodborne diseases and pathogens. Thank you, CHPC. Access to the CHPC has enabled us to produce results as detailed in the following few slides. In this study, 6 Listeria isolates from ready-to-eat meat products were analyzed using whole genome sequencing to identify virulence and resistance genes, prophage sequences, serogroups, and sequence types. Whole genome sequencing identified 4 different sequence types. Further analysis revealed that the strains in this study shared some common known major virulence genes. In general, the resistance profiles for all the isolates were similar and encoded for multi-drug, heavy metal, antibiotic, and sanitizer resistance genes. All the isolates in this study possessed genes that code for resistance to common food processing antiseptics. The aim of this study was to investigate the population structure of Listeria isolated in the meat value chain within the South African market. Based on whole genome sequence analysis, a total of 217 isolates were classified into two main lineage groupings, with the most abundant sequence type 204, 219, and 321 respectively. In addition, 14 clonal complexes were identified with overrepresentation of CC1, CC3, and CC121 in the processed meat beef, ready-to-eat poultry, and raw lamb meat categories, respectively. Listeria pathogenetic islands 1, 3, and 4 were present in 7.4, 21.7, and 1.8% of the isolates, respectively. The findings of this study demonstrated a high level of genomic diversity among Listeria isolates recovered across the meat value chain control points in South Africa. In foodborne disease surveillance, the adage, prevention is better than cure, holds true. The prevention of foodborne disease outbreaks is paramount in public health. The COVID-19 pandemic has demonstrated the critical importance of widespread and continuous testing. The same applies to foodborne disease surveillance. The prevention of epidemics, such as the South African listeriosis outbreak, requires large foodborne disease surveillance and foodborne pathogen testing projects to protect public health and ensure that no lives are lost due to contaminated food. Robust foodborne disease surveillance research projects requires the collection and sequencing of a large cohort of samples, which produces a wealth of raw sequencing data. Typical workflows for foodborne disease research therefore require access to high-performance computing environments due to the extensive data sets used and the computational and memory-intensive applications used in these research endeavors. The CHPC has proved to be a critical partner in my foodborne disease surveillance research projects. I would like to acknowledge my collaborator, Dr. Itumeleng Matle, from the ARC Bacteriology Division, the Department of Agriculture, Land Reform, and Rural Development for the continuous support and funding, and the Agricultural Research Council, and in particular, the CHPC, for making research such as this possible. Thank you.
Well, thank you, Rian, for that very interesting talk. If I truly needed anything more to put me off meat, you nicely provided the evidence for that. We have no questions yet from the audience. Thank you, Evans. We've got Quinn Reynolds in the audience, so we have a question. I'll read that for you. Many thanks for a very interesting presentation, Rian. What is currently the main bottleneck in maintaining continuous surveillance? Is it sampling and data acquisition or the computational resources for the genomics?
Great, fantastic. Thank you for the chance to present some of my work. With regards to the question, I think the main bottleneck would not be the sampling, but actually the generation of data, which is quite costly. Computationally, I think we're relatively well off, but unfortunately, next generation sequencing is currently relatively expensive. And this would possibly be the largest bottleneck currently faced.
Well, thank you for that answer. Any more questions? If there are no more, I would like to pose a question, if I may. And this obviously comes from the side of a CHPC support scientist who works with researchers like yourself on getting these studies completed. Talk us a little bit through what your practical experience of this has been. What are the things that have been difficult, and what has made it a lot easier to get the results out of the CHPC, and what would make it easier still?
So I would like to congratulate the CHPC on giving me a fantastic platform to do everything I need. Probably the biggest issue currently would be the movement of data from where we receive it to the CHPC and then back down again. So with regards to the analysis and all the applications, I've been assisted greatly. Any requests have been quickly addressed. For me, the biggest problem would be movement of data and internet speed and connection to get the raw data on, and then from there to get the raw data off.
Well, thanks for that comment. I think it makes a lot of sense. I think it is a problem that a lot of researchers have. Not everyone has equal access to the kind of networking infrastructure that you need to make the computational world really work. Well, we thank you for your contribution. That was very interesting. We have several minutes remaining until our next talk, and consistent with the way we've been running this conference to allow for any possible attendees from the other session to come and join us here, we will adjourn until shortly before 11:30. We've got about 8 or 9 minutes. Thank you, everyone. Who graduated in 2007 with a PhD in applied mathematics from the University of Cape Town. He joined the CSIR Aeronautics Systems competency, where he became a senior researcher in numerical methods development. During his time at the CSIR, he worked on external aerodynamics, fluid structure interaction, and free surface modeling simulation. He is a co-developer of the Helix Hydro multi-phase CFD package, as well as the open source external aerodynamics solver HISA and the CFD OFCAD front end. He has published 16 journal papers in ISI rated journals and currently works as a senior CFD developer at NGIS Limited. The title of the talk is a multi-physics framework for modeling coupled heterogeneous transport and reaction kinetics. Oliver?
Thank you very much, Charles, and good morning, everybody. Let me just share my screen. Okay, so as Charles mentioned, I work for Engis, which is a company that produces open source software for computational fluid dynamics. This was originally a fork of the very well-known OpenFOAM CFD package. Several of our most senior developers come from OpenFOAM in the days before it was actually OpenFOAM, when it was still foam. And more recently, we've started to use it more as a sort of development kit because our top level software has started to deviate quite a lot from that. But we supply the GPL licensed open source code together with our support contract to industry clients and together with a graphical user interface as well. So the title of my talk is a little bit of a mouthful, so I'm just going to dive right in, and hopefully by the end of the talk, it will become a little clearer. So where this work started from was developing a CHT solver. This was originally developed for electronics cooling applications. The example you see here is forced convection cooling of a blade server. And the thing that was new about this compared to the standard OpenFOAM solver was that we had implicit coupling between the different regions. So in this case, all the different solids, the chips and memory modules and so forth, and the air flowing around them. And that gave us quite a considerable performance boost compared to the standard solver of between 5 and 20 times, depending on the application. So you can see in this little animation, just the comparison in the speed of convergence, comparing the explicitly coupled and the implicitly region coupled solver that we developed. So this was then taken up in the motorsport industry. We added some features like anisotropic materials, which is obviously very important when you have carbon fiber layups, rotating frames, exemplified in this example of a brake disc cooling application. And then consolidated regions, which is a way of handling when you have multiples of solid regions, handling that more efficiently. And that's something I'll talk about in more detail later on in the talk. But it's quite an interesting case in Formula One from an HPC perspective. A couple of years ago, the governing body introduced a rule that every team has basically got a limited quota of computing hours per season. And so it really incentivized the development of the most efficient possible methods. And against that backdrop, a lot of the teams are switching over to open source solutions, which I think really demonstrates that the open source options are no longer just for hobbyists and can really compete in their own right against the traditional proprietary solvers. And of course, for those teams as well as other industry clients, a very valuable part of this is that they have control over the source code and they can integrate it into their processes in whichever way they like. So having done this, we looked at how this could be generalized and came up with this idea of a unified solver framework. So the problem is that OpenFOAM has a huge proliferation of different discrete solvers. Basically, every single possible application, every combination of physics is coded as a separate sort of linear program. So if you look at just single phase flow, for example, you have simple foam, pimple foam, row simple foam, row pimple foam, buoyant simple foam, buoyant pimple foam, buoyant Boussinesq simple foam, buoyant Boussinesq pimple foam, row central foam, sonic foam, all these solvers, and that's before you even start adding multiple chemical species or multi-phase flows. And on top of that, you have several different interfaces for specifying material properties and several, in fact, 4 different definitions of pressure. You have a gauge pressure, a kinematic pressure, a absolute dynamic pressure, and so on. And so for ease of use, this presents a bit of a problem. Each solver has a slightly different interface. It's also quite difficult to know what each solver can really be used for. And of course, there's a tremendous amount of code duplication. So our idea was, starting from this multi-region conjugate heat transfer framework, can we kind of build a modular system of building blocks for the different components that one might like to solve. So you have the flow solution system for velocity and pressure, then energy, then radiation, and then, as we'll see later, chemical species transport. And the idea is that all these things can be slotted together however you like, and that the solver algorithm then becomes not a customized procedural program, but actually a set of dependencies between these different modules. So this obviously improves the maintainability of the code base, gives one much more flexibility to actually deploy applications that haven't been specifically coded for. For example, one could couple flow solution with stress modeling to get fluid structure interaction. You can add dual heating, which is basically coupling electrostatics to look at the heating caused by electric currents. And the list goes on and on. So the first application for this unified solver concept came from a client in the chemical industry, and looking at this structure, which is extremely common in the chemical industry, this packed porous bed of catalysis pebbles. Unfortunately, we can't show the actual example that came from them, but as luck would have it, my colleague Dan Combest had looked at a very similar kind of application about 10 years back, which he tried to model using open foam at the time. And the conclusion from that basically was that there were issues with the meshing of all these complex little pebbles, and there were issues with the solver being explicitly coupled between the regions. The amount of computing time required was really unfeasible. And as you can see from the list on the left, this is quite a complex multi-physics type of problem. So here is what it looks like. You have these pebbles, which are typically made of a catalyst like nickel. They're sort of 3 centimeters in diameter, and these are packed inside a tube like this, typically sort of 20 centimeters in diameter. And air is either forced through or allowed to convect through naturally. Well, not air, but some chemical reactants are sent in, and the catalysts then catalyze this reaction in the pebbles. And the pebbles themselves are quite porous. They have a very sort of serpentine network of little tunnels in them, which means they're too porous to really have any convection flowing through them, but it does allow the chemical products to diffuse into this porous solid, and to have maximum surface area for the reaction to be catalyzed. So the reaction that we're looking at in this case is this Sabatier reaction, which is basically the methanation of CO2, or a way that's commonly used in the chemical industry for producing methane, and that's an exothermic reaction. So what was required on top of our existing sort of heat transfer solver in order to make this possible? For the solid region, we needed to introduce this species transport solver, which is basically derived from mass conservation of the individual chemical species, which has this reaction source term on the right-hand side. Just get myself a pointer, that's over there. And then that also contributes additional source terms into the heat transfer equation. So this all happens fairly seamlessly with the kind of solver module framework that we have. And then we're able to reuse that same solver object on the gas side. You have the same transport of species, except this time you do have convection happening. And there were a couple of missing terms compared to the standard row reacting buoyant foam solver, which is sort of the equivalent that you could use in the fluid region in open foam. And also this allows you to do steady state analysis, which isn't possible with row reacting buoyant foam. And the last ingredient, of course, is a coupled boundary between these solid domains and the fluid domains. Importantly, this needs to be derived to actually ensure proper conservation of the mass flux. Whatever exits the fluid domain must enter the solid domain, and vice versa. Okay, so first step was to do a little bit of verification, and we started with this very simple test case just to check the reaction diffusion system in the porous solids. So we just have the square where we impose a different concentration of the 4 chemical species on each side, allow them to diffuse into each other and react. And for verification purposes, we compared this against the result coming from StarCCM+, which was being used by the company. So just looking at a couple of slices through this square, we show then these as graphs on the next slide. And we got very good agreement, both in terms of the chemical species concentrations and the temperature through these 2 slices. Then the next case for verification purposes was this one, where we just put a single spherical pebble in the middle of a pipe and pass the chemical reactants over that. And as you see in the top figure, you have the methane being produced in the pebble and shed behind it. And then here you see the hydrogen, one of the reactants, being used up, and the temperature which is produced. So the idea here was to validate the coupling boundary condition compared to StarCCM+. Unfortunately, we couldn't do that because it turned out the way that this was set up in StarCCM+, involved basically having to do a sort of a custom mapping at the boundary and set up a temperature mapped boundary condition and species mapped boundary condition. But unfortunately, this is not actually derived to maintain the fluxes, the mass fluxes and the heat fluxes. And so it turned out in their workflow, you were actually generating a large amount of spurious energy at this interface. So that comparison, unfortunately, became a bit meaningless. But we at least were able to look at the chemical balance and the heat balance in the system and ensure that everything was in balance. So returning then to my colleague's case, and just showing you the complexity of the meshing here, this is really where the high performance computing becomes important. We're generating around 100 million cell mesh in these kind of very complicated spaces between the particles. The mesh has about a 0.1 millimeter thickness at the boundary of these 3 centimeter pellets. And so really due to the complexity and intricacy of this, the meshing is quite computationally intensive and ran for about 5 hours on 560 cores, which is more than you'd normally expect for this size of mesh, but just because of the complexity here. The solution running for 10,000 iterations, which is probably more than really necessary, that took 22 hours for a steady state solution on 560 cores. And here, just to show quite superficially some results, if we look at the animation on the right, shaded is the methane concentration, and you can see that as we move from the bottom to the top with the airflow in this domain, methane is being produced is building up in higher and higher concentrations. Conversely, looking at the temperature, you see the highest temperature at the bottom where the reactants are at their most concentrated, and as they get consumed, that slowly goes down. Then just looking at the temperature profile here, you see a phenomenon that's a little bit different to what you'd expect for normal plug flow through a pipe, where you expect the fluid to basically stick to the walls at the sides. Instead, you see this wall channeling phenomenon, which is characteristic of these porous packed beds where you have an effective greater void fraction next to the wall. And then it's just quite interesting to see some slightly unexpected behavior on the kind of detailed level. For example, some of these pellets here in the turquoise region, towards the edge of the pellet itself, you actually see a dip in temperature compared to the surrounding air, which at first led me to believe there was a bug in the code, because that would suggest that you actually have heat fluxing into the pellet when in fact it should be being produced into the pellet. But if you really look at the heat balance carefully, you see that the heat that is being transported out with the outward mass flux actually compensates for that, and you do have a net flux of heat outwards. So those are the sort of detailed things that one can look at in these sort of CFD calculations. And here you see a slice through this, looking at the mass concentrations of the two reactants on the left and the two products on the right. And you see that in fact the reactants don't diffuse terribly far into the pellets, and the products basically build up inside the pellets, and there's a bit of a barrier to diffusion in and out of these pellets. And just for interest's sake, if when we're doing this in a design, we took one pellet here and reduced its tortuosity, so made it easier for things to diffuse in and out. And there you see that you have a much easier diffusion of the products into it. So this is the kind of virtual prototyping thing that one can very easily do with these CFD tools. So now I just want to sort of take a detour into one of the interesting aspects of this kind of problem from an HPC point of view, since we are at an HPC conference. And that is that in these kind of cases, you can easily have hundreds of different solid regions, each one of these pebbles. And the same thing you commonly see in other examples. In this particular case, it's not such an issue because all of these solid regions can actually be merged into one and just treated as if they were a single solid. But in other examples, that's not the case because the current framework basically requires solids with made of a different material to be kept as separate regions. And that's something that's quite sort of ingrained and baked into the software as it is now. And so we do need to be able to deal with these many independent regions. And a colleague of mine has recently done an electronics cooling example of a radar system that had about 300 different solid regions. So this is just a little sort of pictographic representation of what I mean here. We have several different blocks which are representing different solid regions, and each of these is colored in 4 colors to represent a kind of balanced domain decomposition. So that's what happens if you take each of these solid regions and domain decompose it separately. And if you do that, you get very, very poor scaling performance. So this is an example of a case with 120 different regions. Total size of all the solids about 4.6 million cells. And as you see, as you go up to a large number of processes, the scaling simply flattens off and you don't get any increase in speed. And the situation looks even worse if you were to take out the nice big 20 million fluid cells that we have around these. If you just looked at the solids on their own, you would see that it actually gets slower as you add more processes. And the reason for that, of course, is as in this little region here, you're dividing quite a small mesh into very many different domains. You just end up with all surface, all communication, and very, very little interior, in other words, where the computation is actually done. So what we should be rather striving for is the kind of performance you get if you were to just lump all those different solid regions into one single region and decompose that. And that's what's just represented here. And if you do that in that example case with 120 different regions, lump those 120 regions into one, then the scaling performance you get is shown in this blue line here, which is much closer to what you'd expect for this kind of CFD application. You get reasonable scaling until you start getting down to around about 20,000 cells per core, and then it starts falling off. So that's what we ideally should strive for. And the question was how to do this. So the most obvious thing to do is to treat all these different solid regions as if they were one. But to do that, we would need to have some kind of multi-material model where you can specify a different material model for each of these solids and deal with them in one. And the problem for us was that this would be quite a big refactor to the code base. And particularly for the graphical user interface as well, everything would have to change, and we didn't really have the time to look at that. The other option is to say, well, why do we actually solve these regions independently in sequence? Shouldn't the code just be refactored to treat them internally as if they are one thing? But that's an even bigger refactor, because basically every loop that goes over cells in the mesh now has to be aware of multiple different regions. So we had this interesting situation of wanting to achieve something that seems conceptually quite simple, but the obvious solutions to it would have required great amounts of rework to the code. So what the solution we came up with was sort of a creative use of threading, which I'll try and describe with some little diagrams here. So in the case where you take all these different solid regions and separately decompose them, this is the sort of control flow you have. The red and the green arrows refer to different processes in your decomposition. In region 1, they start off in region 1 and both do roughly the same amount of work because we've done a balanced decomposition. Then they reach a point where they have to synchronize and communicate some data via MPI, and then they continue to the next region. In fact, there are really many of these synchronization points in each region, but we don't need to worry about that. So what we would rather do, as we showed before, is just decompose all these individual solids as if they were one. But of course, the problem, if we do that without doing anything else, is that you lose the load balancing. So in region 1, processor 1 has a lot of work to do, processor 2 does virtually nothing, and then sits around waiting at the synchronization point for processor 1 to catch up to it. So the idea then came into saying, what would happen if we could somehow, in processor 2, kind of teleport straight through to region 2 and do this work that's waiting to be done, whilst still waiting for processor 1 to reach the synchronization point. And this synchronization point can be very deeply embedded in several sub-routines. You don't really know when the code is going to call an MPI function. So it has to be something quite sort of generic. So the idea we came up with was to use threads. It's the obvious answer when you need to get from one point in the code to somewhere totally different. But to run them in series instead of the traditional parallel processing paradigm. So basically, processor 2 would do some work. When it hits this point where it needs to synchronize with processor 1, it would just switch to another thread, which starts doing some work in region 2, and so on until both processors have finished all the work up until that synchronization point in all the regions, and then they can synchronize in sync here. So that basically gives us an effective consolidation of all these regions without having to go through all the refactoring in the code. I've never heard of threads being used in this kind of synchronous serial way before, but I was talking to a colleague yesterday and he told me that there's a recent addition to the C++ standard in C++ 20 called coroutines, which he reckons basically does exactly this. So that took the wind out of my sails a little bit, but let me tell you about it. So when we do this using the threads, the initial answer that we get looks like this in terms of scaling. So that was a bit of a disappointment, not nearly as good as we would have hoped compared to the actual consolidating of all these regions into one. In fact, the initial implementation used OpenMP, which was even worse because OpenMP turns out to use busy waiting, which basically is the exact opposite of what we want to do here when we want to run all the threads in series. But it turned out we were able to, this initial implementation was fairly inefficient. I had thought that switching between threads was an extremely lightweight process, but it's actually something that takes about 10 microseconds or so. And so you want to minimize it as much as possible. And doing that increases the efficiency of a fair amount, and we get to this yellow line here, but still quite far away from the ideal. Then a final optimization, and that is to just use as few threads as you actually need in each processor. And this really starts to come into its own at the higher processor counts. where you actually end up having very few regions involved in each processor. And then you start getting scaling, which is much closer to the ideal that we can expect here. There is a little bit of overhead, but that is actually to be expected for other reasons that I won't go into. So in summary, these kind of multi-physics problems are a nice grand challenge. They can allow for virtual prototyping, inspection of models in fine grain that wouldn't necessarily be possible with experiments. In some of these sort of situations, you actually can't do experiments at all because of the hostile environment. And basically, we're trying to make this more practical with this a unified solver framework. And I think, as I've also tried to show in the last section, that in this context, there can be some very interesting additional challenges to running these efficiently on HPC. So thank you very much. I'm happy to answer Professor Reynolds' questions and any others that there might be. Thank you.
Thank you, Oliver. Yes, we're waiting for Quinn to ask his obligatory question. There it is. Thank you, Oliver, for an amazing presentation, and certainly I agree with that. As always, in your unified solver framework, do you have methods to account for motion of the individual pellets in the bed, and also size and shape changes? This could be very useful in cases where the particles in the bed are also participating in the reaction process.
Yeah, very, very interesting idea. I think that this is something that could be put together with the tools that exist in the OpenFOAM framework. It's not something we've actually tried yet, but just off the top of my head, I can't really think of a reason why it wouldn't work. Basically, use some of the existing, for example, 6DOF motion functions and the dynamic mesh functions. Well, I say it wouldn't work in principle, but for packed beds like this, it would be quite a problem because you would have to have contact detection, which we don't have yet. And obviously the mesh deformation wouldn't really work when you have close packing like this. We do have other means of being able to do that with our generalized internal boundaries. So yeah, an interesting problem to look at, but would require a little bit of work.
That sounds to me like easy to fix. You just give in just some money. Are there any further questions? No hands raised. Well, thank you very much, Oliver. That was a particularly interesting talk from my perspective, at least. And thank you to all our attendees. Sure. And we're adjourning now. Thank you.
We shall be starting quite soon. So hopefully you're all online now. Okay, good afternoon. I'm Anton Lopez. I'm chairing this session. Welcome to this afternoon pre-lunch session. I don't believe I've met either of the speakers before, but virtually as we're meeting today is a good thing. Our first speaker is Dr. Lucy Kirui. She is a lecturer at Kenyatta University in Kenya, and her work is mainly focused on chemistry, but has expanded more as well to computational biophysics. Today, she is discussing a biomedical study relating to COVID-19. So, Lucy, you can go ahead and try and start with your slides, and I'll confirm that they are working.
Thank you, Dr. Lopez.
You're welcome.
Let me start sharing. You can see my slides.
Yes, looks fine to me. I'm going to go off.
Okay. Good afternoon. And again, thank you for the invitation. to the 2020 CHPC National Conference. My name is Lucy Kiruri from Kenyatta University here in Kenya at Chemistry Department. And yeah, I'm in Chemistry Department. So my work is more in chemistry related researches. So my talk will also be on COVID-19. However, I a bit changed the title to look at an overview of some of the work that we do and where we come from and the way we came into the CHPC. In my group, we do many studies which are related to computational chemistry. We look at small molecules. For example, in one that is an organic compound, which is a natural product called baracol, which is well studied here in East Africa and also in Thailand. And we have a keen interest. And more of it, we are looking at the stability of this molecule. Since after a short while, it reverts or it converts to anhydrobaracol, it loses water, and therefore we want to understand its chemistry. We have another research which is on photodynamic antibacterial chemotherapy, and this is used for water decontamination. That is to kill antimicrobial in water using porphyrins. And therefore, we're doing some studies. Also, that is ab initio study and also the TDDFT study. We also, in the group, do molecular dynamics. We have a student, a PhD student, who is working on the heterogeneity of an ODBP, that is the odorant binding protein, looking at the various dynamics of this protein to understand on ways of malaria. We also do simple folding and activity for some proteins. I took this slide because I want to explain where we came from. And this here is 2013 at Kenyatta University, whereby we didn't have computational chemistry was new. And this was the start of computational chemistry here in Kenyatta, whereby we were lucky to have some donations of workstations from Novartis, US. And we have these two students who later graduated with MSc, not under me, by my colleague. They were, as you can see, they are very excited once these donations were done. And also a program, Spartan, was also given. And they were able to do the MSc research on TB drugs. However, the challenges that we were facing at that point was that the workstations, since they were workstation, I think they were small, the capacities were very small, and therefore they were slow. Also, it meant that we could not add more programs to the workstations, and therefore the work slowed for the computational chemistry. And later, after joining, this time I had not joined, but later after joining Kenyatta, that is when I applied for allocation at CHPC, and I was lucky to be allocated some time, time for computing. And let me say we are very grateful to CHPC since we were able to expand our computing. We were able to add more students to the group and also have access to very many resources such as program softwares and so on and so forth. So that is part of our success story there. At KU, with current, the cluster is CHEM 1032. I have 6 students so far. And so far, I've had 2 MSc students graduating and 1 PhD student graduating. And the softwares that mostly we use is GROMACS, that is for molecular dynamics. I'm also happy because I requested for Plumed to be added to GROMACS, and this was done. So I'm grateful for the support team at CHPC. And also we use Gaussian. We've used Gaussian 09, and now we are also using Gaussian 16. For the computing challenges, let me say, as we don't complain, we have none. We have 3 students who have graduated, and we have 2 publications. So far, one actually is related to the SARS-CoV-2, and this is on East African natural products and some of FDA approved drugs, which is work was done with collaboration with University of Dar es Salaam, colleagues of mine in Tanzania. So we work together for this paper. And also the second paper is also done using CHPC resources. So again, thank you. We are great for the CHPC support team. Here are some of the students. The first student, Katana Chengo, has graduated, and he did work on DFT studies of the radioselective reactions of alkyl, aryl, and amine substituted pyrazoles with the Lewis acid ion half sandwich. And these systems were very heavy, let me say, and he was able to finish his PhD in 3 years because of using a CHPC. Since we have the ion there, and which is a metal, and you require a lot of time for the DFT calculations. Basically, he used Gaussian 09 and also 16. I have another student who goes by the name Wycliffe. Though Wycliffe has not finished, he's using GROMACS, but he's still running. He's still working on his PhD. Another student who is working on porphyrins. He's a MSc student. Also has not graduated, and hopefully he will graduate maybe this year. My colleague at University at Nelson Mandela Institute of Technology here in Tanzania, Dr. Daniel Shadrack, we work together. He's also in my group. Also for him, he does computational biophysics, and that is MD simulation, mostly of natural products against the SARS-CoV-2. Again, he uses GROMACS and also Plumed. So my talk today, basically, I want just to look to highlight on one of the work that I'm doing. Is done in our group in collaboration with 2 other universities. Actually, one of the universities is in South Africa, whereby we are using synthetic peptides, and we want to use the synthetic peptides for interaction or for blocking the SARS-CoV-2 virus. And so we all aware of the coronavirus, but coronavirus has been there since 2003. The first one was SARS-CoV, and you can see that the origin of these coronaviruses are from the bat, and they get to human through various animals. So the first got through the human through this animal called the civet. It is a cat from the cat family. We have the mouse COM, which was later, I think in 2008, which came through the camel. And for the 2019, it has not been established yet how it got into human. So our interest is to study if we can use the synthetic peptide. And the good thing about synthetic peptide is that these are proteins, and therefore they are non-toxic. So that is mainly the objective of this study, is that this is the SARS-CoV-2 virus, and you can see that it has the spike proteins, and this spike protein has a very great affinity of attaching to the human ACE2. So the virus passes through the spike protein to the ACE2. That is where the binding occurs. And so our aim is to try and block this position here. That is where we want to block using the spike, using the synthetic peptide. So this is the protein itself. It has already been elucidated. I think this came out in March this year. This is the SARS-CoV-2 spike protein receptor binding domain, and this is the human ACE2. And it is believed, or it has been shown by research, that the virus passes through the spike protein to the human ACE2. And here is the PDB, in case you want to look at the protein more. And so we want to establish if we can be able to block this area here, so that we block the protein from entering the human. So we are using synthetic peptides, as I've said, and these synthetic peptides, they've been designed. All of them have not been synthesized in the lab. However, they are designed in such a way that they are antiviral. They are antiviral peptides. And the first assignment, we still haven't named them since they are new. These are new synthetic peptides. And so I've just given them generic names like peptide 1, pep 1 to pep 8, which is peptide 1 to peptide 8. And the first thing that we did was to establish actually if they have the antiviral activity or their antiviral potential through this website here, which is a free website, and you are able to predict whether your peptides are antibacterial or antimicrobial or antiviral. And therefore, you can see that from peptide 1 to 8, they showed a certain degree of antiviral activity. The good thing is that peptide 5 had the highest of 91%, and also peptide 6, 7, 8. And we had to reach at this point and argue that whatever is greater than 70% will be considered as promising, and therefore we can continue. However, this is not very conclusive, and we went by to do some MD calculation docking experiments. So MD docking experiment was again done using this protein, which is the SARS-CoV-2 chimeric receptor. And as you've seen from the previous slide, it is complexed with the human ACE2. And again, here we used a cluster from the MIT supercomputer. It is called the ClassPro 2.0 server, and we used this to do the docking. So again, it is a free software. And from the docking results, here are the docking results. So the one which appears as purple, this is the spike protein. The cyan, or the like blue coloration, is the human ACE2, and the red is the synthetic peptide. And you can see that for peptide 1, the synthetic peptide attaches at the end of the assay 2. And the same case applies for the PEP2. It attaches at the end of the assay 2. Not where we want it. We want it to be broken here, but again, they attach to the end. And this binding was similar for peptide 3 and peptide 4. For peptide 5, we can see that the synthetic peptide attaches close to where we want it. That is at the interface, that is between the spike protein and the human acetyl. And same case for peptide 6, whereby it attaches in between, okay, not really in between, but close to where we wish it to be to block the virus from entering the body. And again, this was similar for peptide 7 and peptide 8. And remember that peptide 5, 6, 7, and 8, they showed very good antiviral activity. So this work, again, is very promising. So however, again, one will argue this is not conclusive until we see the energies. And here is the table of the energies. So we can just look at the column with the lowest binding energies. These ones are too busy. We may not opt not to look at them. They are only the hydrogen bonds and the hydrophobic interactions. But the chemists, we all care about the energies. And from here, you see that peptide 3 gave us the lowest energy, which is minus 850.7. However, we can remember that it attaches at the human acetyl, but it gives us the lowest energy. So that is also key, and we need to also think about it. However, also, we were interested with 5, since 5 has the highest antiviral activity of 91%. It also attaches close to the interface. So these two, we carried them further for MD production, or for molecular dynamics analysis. So these are the hydrogen bonds. I mean, just where the synthetic peptide attaches with the protein or the human acetyl. Similarly, in these are the hydrophobic interactions. Hydrophobic interaction, we get this from the leaf plot. Hydrogen bonds, you may get this from the PyMol program or from the leaf plot. Either of the two will be able to get those results. So these, I won't talk about them. These are just the hydrophobic and the hydrogen bonds from the leaf plot analysis for PEP1 and PEP2. I've not showed for all of them, and this is PEP5, which occurs at the interface. So this is one interface, and this is the other interface. This is the protein, and this is the human acetyl. Similarly for peptide 6, and so on. So here are the basic analysis for this work. So we have peptide 3 and peptide 5. That is what we carried along from our preliminary work. And we can see that from this, the potential energy, we can see that there is a good stability of the potential energy. Just after 700, this one is after 500 picosecond, after 700 picosecond for the PEP2. So we can say PEP2 took longer before it stabilized, but again, it stabilized at 700 picosecond. So we can say that is a good steady convergence. Again, when we look at the temperature, which the temperature, we've set the temperature at 300. Again, we can say that the temperature have equilibrated very well for PEP3 compared to PEP5. PEP5, there is a lot of up and down, but this is expected since this synthetic peptide is at the interface between the spike protein and the human ACE2, and this could be causing all this rambling in energy. But however, we can say that technically it has converged at that, although it might require maybe an additional run of that temperature to see if there was any problem. If we look at the pressure, again, pressure at 3 is still very well, again, compared to 5. And this, again, is because of where these peptides are attached. Although after maybe longer runs, this will be clear. So the density is okay. The density is again steady for all of them. Not the same. We have a lower density with PEP5 compared to PEP3. I know the expected value for this experimental value should be 1,000, but again, in MD runs, we expect sometimes changes with the density. But however, comparing the density and the pressure at the same time, we can say that the protein is stable, or the interaction between the proteins and the peptide is stable. Okay. So right now we are having this work. We've not finished. We want to accomplish 200 nanoseconds. The work has been slow a bit. We've done 150 nanoseconds. This is what we are getting. Although we are using just one node, the performance, since it's a big protein, it's a huge house, we are getting 9.7 nanosecond a day, which is okay. And maybe in the next 1 week or so, we'll be able to complete to 200 nanoseconds, and we'll be able to share results with the world. So that's all for the work. So I'm grateful for the chance to share with you what we do here at Kenyatta. Thank you, and Gia Boga, don't know whether I pronounced it in the right way. CHPC, this work is actually done on your platform, and we are grateful. I'm also grateful to ICTP, the International Center for Theoretical Physics, where I'm an associate there. And also this work has been, we have a small grant from the Kenya Education Network. So I'm very grateful. Thank you.
Thank you, Lucy. We seem to have one question from my colleague Krishna, or just a comment. Of course, we got the quintessential question as well. Okay, but let's start with Krishna. Just a comment. If you wish to get better performance for GROMACS, you should run it on the GPU and not on the CPUs. Yes, GROMACS does run better on GPUs.
Thank you.
Krishna can advise more on that because he's actively involved with the GPUs. And
yeah, I'm grateful because, thank you for that.
Sure. And then we got the quintessential question from Quinn Reynolds. So thank you very much for a very interesting presentation, Lucy. Are you planning to move forward and commercialize any of the new drug therapies that you have identified here?
Yes, especially for the peptides. The ones that I've showed as peptide 5, that is why we are not showing the names. If it works very well, we're gonna synthesize it, and that's our next stage. Hopefully we can be able to do that. Yeah.
Okay, that sounds good. Thank you. Thank you for coming, and
Thank you for the opportunity.
A long way from Kenya, but anyway, it's virtual. And thanks so much. And I think we shall now go. We seem to be running quite fairly on time, so we may have to wait another minute or two before Ron starts. So does anyone else have a comment or question or anything in the next minute? Otherwise, we can just hold on till that, because we need to make sure that they're parallel sessions so that we don't, people can join at the end of another session. Okay. Thank you, Lucy. Does anyone have a comment or anything? Let me just recheck if there's anything more on the Q&A. No. So if you prefer, you could shut off, or if you want to stay on, that's also fine.
I want to stay on for the next talk. It's okay.
Yes, I mean, stay on with your video and your audio. Oh, there's a Q&A. There seems to be a question. Okay, let's see. From Afra Karala. Thank you very much for a very interesting talk. Are you considering any future collaborations?
Yes, I welcome them.
Okay, so maybe you should get in contact with Afra, or she can get in contact with you, or via Krishna. Krishna, Krishna should know the.
Yeah, Krishna has my email address.
And he probably has. And the other, yes, and the other colleagues. Okay, I think we should actually go to Ron now.
Okay.
Hi, Ron.
Bye-bye, and thank you.
Bye-bye, thank you. You're welcome. Thank you. Okay, so Professor, welcome, Prof. Ron Machaka.
Thanks.
You are a material scientist and a WITS graduate, and now a senior CSR researcher. And Ron is today presenting some machine learning work related to high entropy alloys. So without any further ado, I think Ron can continue. It looks like your slides are working.
Okay, great. I hope I'm sharing the full slide, not the one with my notes and the next slide. Can you confirm that?
I can see a slide with the title and then the CSR, Science and Innovation at the bottom. Is that
okay, fantastic.
The full slide? Okay, cool.
Okay, great. That's awesome. All right. Thanks so much, and thanks so much to the organizers of the 2020 edition of the CHPC virtual conference. The work that I'm going to present is is work that started off as a small bottom drawer project, and so I'm presenting it for the second time, but today with all the results. So I'll start. So this is the outline of this presentation. What I will do is that I will talk just a little bit, maybe in one slide, the work that we do at the CSR, the kind of stuff that we actually get paid for. And then I'll go into these materials that we call the high entropy alloys. I'll introduce them briefly, use a couple of examples to say why exactly do we want to work there, and identify some of the problem areas that we think that we have identified with these materials, and the data now that we collected for the machine learning, and some of the methodologies that we use there. Go a little bit into what we got and what we think it means, by way of discussing the results, and then lastly I will talk a little bit about the work that is currently going on which which is future work in this particular case. So, we derive most of our mandate, or we derive our mandate for the projects that I'm currently working on from the Department of Science and Innovation. And what they require us to do is to localize manufacturing capabilities, develop new products, and so you can just begin to realize that, I'm a through and through, experimentalist. So by way of example, we realized that there was a lot of 3D printing that was required in the country. So we developed a new process for converting materials that are not suitable for 3D printing to make some spherical powders that can be used for 3D printing. So these are actually our results. And, this is 3D printing, and this is a project that I've worked on for quite a number of years called metal injection molding. And, we were very fortunate that, the guys at WIPO and the guys at the British IP office thought that this was patentable. So we do have a patent that has been filed. It has gone through quite a number of hoops, and we are looking forward to the outcomes. For this work that we have done here, we're also very fortunate that we got an outstanding contribution by a research team at the 2020 CSIR Awards. So this is the work that we do for the CSIR, for the Department of Science. The work that I'm going to talk about today is, there's a new group of materials, a class of materials that has come through. These materials are called, high entropy alloys. And these alloys, are special in some way, and I'll explain why. You see, in this slide, as you realize, the slide goes back into history. If we can go as far as maybe the Bronze Age, if you allow me, geez, I'm so sorry I was not there. So this is not first-hand experience, but what we know for certain is that all the way up to maybe the World Wars, most of the alloys that you get, if you're looking at the chemical compositions, if you're looking at the structure of these alloys, you have maybe about two principal elements that are in there. So if you're looking at maybe some steels that came up, then maybe we are looking at iron and carbon. Some of the early aluminum alloys, it's maybe aluminum and magnesium. So that's what we are pretty much looking at, all the way up to maybe the end of the Second World War. But the Second World War did something else, because you start getting some of these exceptional, for high performance materials, some super alloys come in, some high strength stainless steels, some very good aluminum alloys begin to come in. So this is just at the end of the Second World War. Then these things, they become, something that is researchable. And what you get there is that, the chemical compositions begin to increase just a little bit. So that's what this blue line means. It says that as time went on, the complexity of the materials that we are dealing with, it was just becoming a little bit more on the, in the exponential side. And then we come to these high entropy alloys. Around 2004, quite a number of two papers came out, and they said that, listen, you can move away from the idea of having 2 or 3 elements in there. You can pump in as much as you want. And then you have this new class of materials that relies mainly on disorder, which is really awesome. If disorder can give us wonderful properties, but something else happens. As we move into these high entropy alloys, the alloys become multi-component. They become more complex. From an experimental point of view, where trial and error was okay before, it is not okay now on the high entropy alloys, because the number of combinations, they have also skyrocketed. Where pencil and paper was fine before to generate data, now it's becoming fastly and mostly impossible. As you start adding in some of these expensive things that you get in there, the hafniums, and some of these very exceptional, exotic elements, the cost of doing an experiment skyrockets from somewhere like 10K to hundreds of thousands. And maybe one might ask, if we have identified all these problems and the headaches that comes in with the high entropy alloys, why are we going there? Because stainless steels work. Bronze, in as much as it was one of the first, in the alloys, why are we spending time doing that? Then questions can be asked. And I think this slide explains that quite a lot. And it explains it in two different ways. If we are looking at the strength of a material, whether this is cracking or deformation, you begin to realize that concrete is down there, and then we have some of these high entropy alloys that are sitting up there. We know that, as humans, we have ambitions. We struggle with weakness. So we want everything else to be stronger. We want things to be lighter. We want things to go faster. We want things to be better, go further, last longer, or be dope if you're a little bit younger than me. That's what we want in most materials. But you see, as we do that, then it says, yeah, but these things work. But if you give that as one example, we are not talking about ESCOM. This is just as an example. The COO of ESCOM in 2019, this is from 2019, they said that leaks are biting us big time. So weakness is something that we cannot tolerate in our environments. So just to give you an example of what high entropy alloy is. So what we're just doing there is that we're just pumping quite a number of different elements into a particular structure. So if this is titanium, we can replace some of the titanium atoms in the structure with let's say with nickel. And then so this is titanium nickel, and we know that it's still a traditional element. So we call this traditional element. But then if you go and pick aluminum, cobalt, hafnium, and then other things, and then you toss them into this structure, then this structure becomes a high entropy. It becomes a cocktail of elements. It becomes a high entropy alloy. And as you can see, it's slightly distorted. There's a lot of disorder in this structure. The entropy of the system has skyrocketed. So that's what, but why would anyone want that? So if we give these, these things a little bit of, introspection, you realize that if this is titanium, we know that titanium does not have what is known as a shape memory effect. The effect where a material remembers its own shape when it's deformed and heated. If you look at the titanium nickel alloy, it has a little bit of this shape memory effect. It's a traditional alloy. We know that over a small region, maybe between 30 degrees and about 90 degrees Celsius, it remembers its shape. So it has a limited shape memory effect. But as you take advantage of the cocktail effect and add in a little bit more of what you can add in, we can increase the window of operation of these shape memory effects or shape memory alloys all the way from -100 degrees to about 100 degrees Celsius. So this is one of the reasons why it is important, or why it is something that is worth pausing and looking at these, these new materials. An experimentalist, so so we picked it up very quickly because we have different atoms in this structure that are randomly sitting at all sorts of lattice sites. It creates this cocktail effect that makes a very good recipe for confusion. Now that's not my words. It's in one of the one of the serious books on. If you look at the type of alloys or the type of elements that can be used to make up these high entropy alloys, you realize that this is across the periodic table, and there are thousands and thousands of possible systems that can come out of these combinations. All of this, what it does is that it creates overwhelming complexity where we say experimentalism is beginning to fail, or pure experimentalism is beginning to fail. So in the complex corner where we wanted to operate so that maybe we can solve ESCOM's problem, we now quickly realize that there is a price to pay. But because there is a price to pay, we also realize that doing experiments alone is not the solution. So it doesn't come out very nicely. So the work that I'm presenting today is to try to answer a very nice question, which says, because we know that the reason why these high entropy alloys have got exceptional behavior, exceptional properties, we know that it is because of the phases that are formed. So it's extremely difficult to do the experiments alone. So what we have decided to do in this particular case, we say someone has done the experiments, because there's literally a paper that comes in pretty much every week talking about these things. So there are thousands and thousands of results that are out there. Let's collect those ones, put them in a database and run machine learning so that maybe we can limit the number of experiments that we can do. So what I did was that I went around and collected about I think now there are almost 1,600 entries and created a data set. This data set has information on the elements, it has information on the properties, it has information on how the alloys were prepared. And so the reason why this is a little bit on the special side is that we collect both the phases and what we call here metallurgical features, which is information relating to how they were prepared as well as the elemental. And then we created a new data set for this. And we think that it's one of the biggest out there. We have been sharing it in bits and pieces with other people, but it's one of those things. So, oops, did I just? I think I lost my net. Hello, can you guys hear me? Okay.
I can hear you, yes.
Hello? Oh, okay, great. Okay, so in this particular slide, I show what the framework that we proposed, because so many different phases can form. So by phases, we are saying that, for those who are familiar with, a little bit of metallurgy, any material can, can take quite a number of different structures. Either it can be, a cubic structure, which is body-centered. You have one atom that's sitting at the center. It can be face-centered, where you have atoms that are sitting on the faces of the structure. It can be hexagonal. It can take, or it can take combinations of both. So we collected all this information. We processed the information by removing some of the phases that we thought maybe we're going to introduce some, some noises. I'll explain that, briefly. Filtered, the information, and then we ran machine learning. So the way I speak about this might not be that great for other people who are, in the field. So what we did is that we split the data set into two, 75%, 25%, and then we trained the models. But what we did there is that in training, because we realized that we had so many features in our data set, we wanted our feature selection. What we wanted to do was that we realized that there was a problem where if you use maybe random forests alone to determine your most important feature, we realized that there was a problem that was coming in, because the more feature selection criteria we used, the different results we got. So we decided to run something else that we called a majority vote. So each feature selection, experiment that we ran, or implementation that we ran, it was selecting its own ranking system, and then from the ranking system, we combined those ranking systems, and then they each voted for what was supposed to be, the best majority feature. I can tell you this became a serious problem when, when we sent the paper for review. They accepted the concept at the end of the day, but it became a serious problem. From the best feature system that we had, then we did the multi-class classification. and then we were able to do the rest. So the implementation, just this is just to focus on what I've talked about. So for the multi-class classification, we used Boruta. We used the random forest based recursive feature elimination. We also used SVM, and then we also used LVQ. Then the majority vote is really to use these feature selection algorithms to vote for the best systems. And just for the fun of it, every vote was supposed to count. The multi-class machine learning classification, for phase formation, the algorithms that we use the decision trees. So we use all 8 decision tree, LDA, naive bias, generalized linear regression, random forest, and the nets and other popular ones like the k nearest neighbors, as well as the support vector machines. I know that there are other classification algorithms that are there that are more powerful, that are the the boosted ones. But I did not think that maybe I understood the work that much, so that maybe we can venture into those ones. So we use these ones that we could easily understand, so that we can understand what's there. We did the normal things, for data preparation, for this. And these are some of the tuned parameters that that we got from this work. Okay. So as with anything else, it is always nice to have lots of data, have lots of features, so that we think that maybe it can be able to to explain things a little bit more. But what we quickly realized is that on the on the outcome feature that we that we had, we could have a BCC structure, an FCC structure, or a combination of BCC and and FC. And then there are some intermediate phases that form, because these structures are so complicated, or are so are so distorted, and the entropy is so high. You can form other intermediate type of phases. So we call them IM. But what we saw with these intermediate features is that when we started running this, we realized that our accuracies were very low. And consulted a little bit. There's a prof at Prof Wang at at UJ. I talked to him just a little bit, and he indicated that maybe the IM was was the biggest problem. So we removed the IM classification, and then we were able to push up our accuracies a little bit. But what that did was that it limited the the study only to these super solutions, solid solutions. This is the ranking structure that we ended up with, and a puzzling thing was that how we process these alloys was not very important. I know that my metallurgy friends will not like that, but but that's what we got. We tried very much to do everything, quite a number of times, using different techniques, and then it came to exactly the same outcome. So to try to visualize, what we did is that we ended up grouping the features and then running different classifications. And the accuracies that we get, we find that about 13 up to about 13, that's where the top 13, they give the best prediction. Beyond that, we are wasting computing power. To compare these results with what others got. So at the bottom here, this is what we got. Compare this with what others got. The most important thing for us here was our feature selection methodology. We thought that was supposed to be a little bit on the special side. And we also think that the data set that we use is a little bit different, because it's refreshed, it's using only experimental data. So for my friends who do DFT, I'm terribly sorry for that. In terms of accuracies, we got, as good as, about 97.5%. With the random forest, it gave us, the best results. The SVMs, they gave us the best. But what you find is that, LDA and, and then the Bayesian things, they were a little bit problematic. They underperformed. But when you looked into literature, we realized that they say that with noisy data, they tend to struggle a little bit. And because we started off with, an ambiguity that comes along with our phases, we were able to, to know that. This, I think, for me, was the most interesting part, where we said, can we now go and find, oh, actually, no, this is not what we said. This is what one of the reviewers asked for. So this is what, under the reviewing process of the manuscript, they said, can you please introduce new data to the model so that the model can be able to predict something that was not there. So we picked, about 5 different systems. I'm going to talk about 3 of them. So in the first system, the amount of molybdenum was changed. And they say that, the system is supposed to remain body center cubic, for X between 0 to 4%. And we were able to, confirm that. So what we did is that, because the paper that talked about this had left a gap. So what we did is that we just introduced 1, 2, 3, the amount of moly, and then we guessed, and then we tested that against the model, and then it correctly predicted that. We did the same for another system with, chromium, going all the way to an equimolar composition. They say that it's supposed to change so that it can become a BCC structure. We were able to confirm that. The last case, which I'm going to talk about, is a little bit confusing because here we're talking about phase changes. And so, we think that the model works. But what these guys are saying is that the reported works are saying that, the structure changes from FCC to being a dual phase FCC plus BCC around 0.6. We get that. But when it changes again, from being FCC, BCC to BCC alone, the experiments tell them that this is happening at 1.25. We are getting it at 0.75. Now, I know that our model is not perfect, but then at the time we wrote the paper, I did not see it, but I only saw it later on that in fact this system here is exactly the same as this system, which was BCC. So there's something wrong with the experiment from this, because this result, the 0.75, we think that it's, I don't know, we think that it's supposed to work. Anyway, that's one of those things. In terms of future work, we have done the first part, which is material discovery. We call it material discovery. We think that with the model, we can now choose a composition and be able to predict a phase. So what we are now doing is that, because we already have the data, and what we are doing now is that we want to go into property prediction. If we can pick a composition, can we be able to predict also its phase and its properties? That is where we are. So the first part we are done. So this is where we currently working. In terms of the contributions, we know that the first paradigm is experiments. We know we have spent a lot of time working there, and we know that our DFT guys are working in the third paradigm. But here in the fourth paradigm, we think that maybe there can be room for synergies. But we think that the contributions of this work is that we have a framework that is a little bit new. We have a model that works. We have a feature selection idea that maybe that it's yet to be tested by others who put it out there for others to test. And we have a new data set that we are willing to share with with our colleague. I might have gone on a little bit too far, but we are not experts here. This stuff we don't know. I was just playing around with some of the things that I was seeing with the information that was available. So I know that there are other people who are more of experts in this area. We need to take this work into the CHPC So, you know, we welcome collaborators. So those are my contact details. And thank you so much for attending.
Thank you so much, Ron, for a very interesting talk, as was Lucy's. Please could anyone add the questions? Oh, there is one has arrived. It's a quintessential question. Thank you, Ronald. Very exciting work. Have you considered combining molecular simulation results with experimental data in your training data sets to increase their size and improve their coverage?
Yeah, I think this is one of the things that you know, that we possibly do. But as I said, you know, when the work started, you know, I was just checking out something that was new, that was around. So it started off as a pet project. I did not know that, you know, at one stage I'll be presenting it at this forum. It was just for me.
Let's see if any other questions arrive. You can continue to comment on anything if you wish to. I don't see any more questions at present, but let's wait a little bit longer.
Yeah. And I think maybe one of the things to, you know, to point out as well is that, you know, because the work is not necessarily the core of our business unit work. So it was actually not being funded, you know, by anyone else. It was a bottom drawer thing that I did. And thanks a little bit to COVID, I had enough time to finish it off.
It's good. No more questions seem to have appeared. We've still got another minute or so. Oh, there we go. Krishna has put in a question. Is there a reason why you chose to make use of R instead of Python for the work?
Yes, there is a reason. It's a selfish reason, because you know, I started doing this work after finishing off. You know, there's a small project that I did for my master's at Viz Business School, and I used R for that. So I think I was brushed up more on you know, on R to handle the data a little bit more. You know, but there's absolutely no other reason. There is no other reason. It was familiarity, really.
Okay, because you were used to the code, yeah. There doesn't seem to be another question. Let's pause for a few seconds and see. I should mention as well that the next session normally should start at about 2 o'clock, and we have lunch after that.
Okay.
I'm still looking if a question appears or not, but it seems like there is none. So we should restart again at about 2, according to the timetable. Is there anything on typing on this side? Let's see. No, let's go to the room. I think we're going to close. Oh, okay. Oh, is it still open? It said something was going to, okay.
Thanks so much.
Yeah, sure. Thank you to our speakers, and thank you, and enjoy your lunch, and thank you for the good talks, both you and Lucy. Okay, thank you.
Yes.
Thanks.
Right,
Let's see. Okay, all the best. Thank you.
All right.
Keep in contact. Hopefully you can, yeah.